So it is my great honor to introduce NIMA MESGARANI. I hope I'm pronouncing it correctly. I think it is. So he is our speaker today. Um, he is at the University of Columbia. He is well known for his research in the theoretical and also on the practical part of how the neural mechanisms work towards producing speech. He has lots of awards. He has awards, for example, in the National Science Foundation. So he has a very, very big curriculum. So let's welcome our speaker.
All right, well, thank you very much for the introduction and also for having me here. It's great to be back. I've been following this workshop very closely from the time I was at Hopkins about 10 years ago, and it's always impressive how much work is done here. And one of the things that's always discussed in this workshop, as long as I remember, is the problem of speech in noise. As you know, it's still a very difficult problem in most applications. And we also do some work in our lab in the domain of speech enhancement and separation. Is the audio okay, by the way? Yeah, all right. Things like the deep attractor network or time domain audio separation that we use for speech separation. But I figured that you probably have heard enough about speech technologies and deep neural networks. And I decided to focus more on a different type of neural network, the wet neural network for the talk today, just to hopefully add to the variety of the topics that are discussed here. All right, so this I'm sure we're all very familiar with here, this loop of that enables the speech communication. You have the brain, there is some electrical signal that's produced in the brain, it's sent to the muscles in your vocal tract. It produces a movement, and as a result, you are producing a vibration in the air. It reaches another person's ear, and the vibrations of the eardrum are translated into another set of neural activity in their brain. And suddenly they know what is it that you want to communicate, who is talking, what's the emotional significance of the speech. So obviously, air is shared between all sound sources. So if there is another sound source in the environment, that would also add to the vibration that the receiver gets, right? But somehow we know that, we humans are really good at this work. And machines are also getting really good these days to do this. So the question that we are interested in part of my lab, in half of my lab, is just the basic science part of it. Like what enables the brain to receive this vibration and separate the sound sources, separate the speech from noise, interpret the speech message, right? And what do we know about that? The short answer is that we don't know much enough. It's a very difficult problem, and when we think about speech communication, we usually have a scenario like this in mind, right? So you're having maybe coffee with a friend, you're having a conversation, and for a long time we've been looking and studying this problem like this, right? You have clean sound sources in a very nice environment, and we want to know how they are processed. But as you all know, the reality is more like this, right? You have street noise, you have other people who are talking nearby, planes are passing, you have a bar close by, there's a bar noise. And especially for someone who lives in New York City, if you look at the noise map in New York City, it's really remarkable. It's impossible to find any location on that map that has a noise lower than 45, 50 dB. Right? So having a conversation in quiet is almost impossible where I live. Hopefully you are better off. All right, so how do we study this in the brain? Well, let's consider two cases where we have some kind of an environmental noise. For example, the jet noise from the NoiseX dataset, right? And of course, the hardest type of noise that you can have for speech is another speaker, right? Because then the overlap between the sound, between the target and interfering will be maximum. So this is the question that I'm going to focus on today. What sort of mechanisms enables our brain to filter noise from speech? And this has to be adaptive, right? Because the noise always changes, and it has to be dynamic. Because one moment you might be listening to one speaker, and then you decide to switch to the other speaker, right? So there has to be some dynamic property to this that changes who is noise, who is signal for you. And this is my one slide summary of human auditory cortex. All right, so we have the ears, we have the cochlear that is basically like a mechanical Fourier transform. It, it decomposes the incoming signal into time frequency representation. And that is sent through multiple stages and ultimately it reaches the auditory cortex, which is the pink area that you see over there. These numbers roughly show the number of neurons that we have in each of these regions. And as you can tell, there is a huge expansion of dimension, right? And, like for this crowd, you might think of it as a deep neural network that, you have these different layers and they keep expanding the dimension of the input representation. All right, so if you want to study this, obviously we need to have a way of measuring how neurons are representing the sound, right? Like what is it that they're responding to? And for that, we need to have a way of measuring the brain activity. Of course, you can do that in many ways. There are non-invasive methods that you can just put electrodes on top of the skull, right, EEG, or you could put the head of a person into a very strong magnet, like fMRI, or just measure the magnetic field around the head, which will be the MEG method. And those methods, they all have strengths and weaknesses. The method that I'm going to focus on today is really the good thing about it is that it enables recording from the parts of the brain that we are very interested in. One of them is right there in orange. So that would be right here on the surface of your brain, which we call the superior temporal gyrus, STG. And you can think of it as a non-primary auditory cortical area. What that really means is that if you have a deep network, this is like way up, right? This is like a very deep layer. And if you look inside the sulcus, you see that green area, which is called the Heschel gyrus, and that's more of a primary area, right? So that is like lower in a deep neural network. Analogy would be lower, closer to the input there. So if you want to record from both these areas, which will be great, right? Because then you have a network and you can record from shallow layers and deep layers and try to understand what sort of signal transformation happens there. What we do is we collaborate with neurosurgeons at Columbia and Northwell Health. And these are undergrads at Columbia. Not really. So these are like neurosurgery patients. These are people who have epilepsy. And as part of their procedure, they are, the surgeons put this mesh of electrodes on the surface of the cortex. Or they drill holes in the skull and they penetrate electrodes into the brain. And this, of course, is done purely for medical reasons in order to localize their seizure activity. But these patients are in the hospital for periods of weeks sometimes. And they are plugged into a computer 24/7, recording the signals. So we get to go to these patients and ask them if they want to participate in our research. And if they say yes, we play them some sounds. And all right, so what sort of activity do we see? Now, this is the cross-section of the brain, right? And those areas that I showed you are now highlighted there. So this will be the primary, like the shallow parts of the network that I told you, and this will be the STG, the secondary, the deeper part of the network, right? The higher level representation. And if you play speech to these people, these are the sort of neural activity that you see. Right? So this person is listening to speech, and you look at each of those dots is one electrode, and you see how the voltage in that electrode changes over time. Right? So there is this sort of representation that basically says something about what's in the input, right? And that's what is it that it's encoded? That's really the question that you're interested in. All right, so this first study that we've done recently is based on a work that I did when I was a grad student, and also many other people have shown similar things to that. That what you see in this case is that's a brain of a ferret, because my advisor used to work with ferrets at that time. And if you play noisy speech to a ferret, and if you reconstruct, right, you just put a filter to reconstruct back the sound from the brain of the ferret. What you see is that the noise is largely removed, right? And as if, the brain is doing this speech enhancement for you, and this is the output of that. And many other people have also confirmed that that is happening in the brain. Like there is an area, there is a level of representation that the noise is largely separated and suppressed, right? So we wanted to know how it happened, when it happened, and where in the brain, in the human brain, it happens. Right? So in other words, we wanted to study the dynamic properties of this process. And obviously, this probably has a good purpose, right? And if it happens, how does it change the encoding of speech and perception of speech? Like, does it make you better in understanding the speech in noise?
Just a quick question. Are you saying that even spread, if, like, I assume it doesn't care about speech, right? Correct. It still filters.
Is it speech specific, you mean, right?
Yeah, is it specific or why ferret cares about speech?
Yeah. So we in this paper we also played ferret speech to ferrets. So these are just ferrets, like talking to each other, and you see the same thing, And we have a simple model in that paper that shows that this probably doesn't require any knowledge of speech. It's basically just it's actually very similar to speech enhancement, that you have a modulated time-varying signal and you have something that is relatively stationary, and there's this simple mechanism that could just filter out what is more stationary from what is not. But it's a good question, like where does it get speech specific? All right, and the other thing that we wanted to study is that so what happens when the background noise changes, right? Because that can tell you a lot about the dynamic property. Like if you're talking to a person and then you walk from the street to a bar, right? So the whole acoustic environment has changed. How do you adapt to that? What sort of adaptive mechanisms we see in the brain? All right, so here's the task that we put together. We have these 4 different conditions that you're all very familiar with. These are from NoiseX database, right? And we have a task that there is a speech story in the foreground and the background condition constantly changes, right? And let me see if I have a sample of that and if I can play that.
It is me again, Hank the cow dog. I just got some terrible news.
Can I send the audio to the projector, to the HMI? Is that a...
Me again, Hank the cow dog. I just got some terrible news. There has been a murder on the ranch. I know I should not blame myself. I mean, a dog is only a dog. He cannot be everywhere at once.
So you hear the background noise keeps changing, right, as you're listening to this story. So what do we see in the brain? Like if we look at the time lock responses to all the situations that the background changes, what is it that we see? And here are some of the examples. So each of these plots are 167 electrodes all over the brain, right? And what you see is their average response every time you went from something to jet noise, to city noise, to bar noise, and to the clean condition. So one thing that you can clearly see is that there is a transient response, right? Every time you go to a new condition, there is this change in the neural data that then disappears. And it changes. Like for some noises, for some conditions, it shows up in some electrodes, and for other conditions, it shows up in different electrodes. If you compare just clean task and noisy task, you see that there is really this adaptation, right? So there is that shift, and then it goes back to whatever it was, as if the noise doesn't even exist in the environment, in the sound. So, this is interesting, but of course you want to know what is it, like what information is coming through, right? Like this transient response, what is it it's encoding about the input? So what we did is we used this method of stimulus reconstruction. And the encoding problem is that you have the sound, you have the speech, and you want to predict the responses in the brain. The decoding part of it is that you turn it around. You say, I have the sound, I have the neural data, and I want to know what the sound is, right? And the simplest way you can do that is basically just to use linear filters, right? You give each of those electrodes a linear function of time and frequency, and by solving this problem, you basically have a very simple estimation of what the input was, right? And that's good enough for this case because we are not trying to get the best reconstruction. We just want to know something about, what information about the input is encoded there, right? Because the interpretation of this is very difficult because you don't know what space you're in. But if you map it to something that's very familiar, spectrogram, it becomes much easier. Okay, so what do we see? So here are the original spectrograms time-locked to the transition to each of those conditions, right? So every time we go to jet, to clean, to bar, and it makes a lot of sense. Jet is a high-frequency noise. You have that there. Bar is more of a low frequency. You go to clean, suddenly everything becomes nice and quiet. Now, what if I do the exact same thing, but with the spectrograms that are reconstructed from the brain of the person? This is what we see now, right? So this is now that transient response interpreted as a spectrogram. And it's interesting because you see that what comes through to the data is really the features of the noise. For example, that high-frequency jet noise, low-frequency part of the bar noise, and this interesting-looking thing that is like a rebound. Every time the noise disappears, you have this rebound in the neural data, right? So, what this really tells us is that every time the noise changes, the noise comes into the brain, and then immediately it's suppressed very quickly, right? In matters of hundreds of milliseconds, there's this adaptive mechanism that suppresses it. So another way of looking at it, that's just the average spectrogram of those different conditions. This is during the adaptation. So when you are in this period, you're still adapting to the noise. You see that they really look like the noise. And after the adaptation is over, like when you are in this part of the signal, you see that everything converges to just the average spectrogram of the clean speech signal. Right? So this seems really like a mechanism that is suppressing the noise and keeping the speech. Of course, you can quantify that. It shows the same, that they become like noise, but then they become like speech. And this is also the timing, right? So that allows you to measure the signal-to-noise ratio of the representation. And you see after the noise changes, there is a decrease in the signal-to-noise ratio, and then it recovers. So, another way of showing this would be to try to simulate that, right? Create a speech signal that has this sort of time SNR over time, right? Then it allows us to hear how it sounds. Okay, so the task that I played you before, now I'm going to modify it based on this signal-to-noise ratio, right? And this is how your brain actually hears that.
Dinner table. Was it my fault the jelly jar fell off and broke?
So this is the original. One more time.
I jumped up on the dinner table. Was it my fault the jelly jar fell off and broke?
And this is how your brain interprets it.
I jumped up on the dinner table. Was it my fault the jelly jar fell off and broke?
One more time.
I jumped up on the dinner table. Was it my fault the jelly jar fell off and broke?
That gives you an idea of the timing of how quickly your brain adapts to speech. All right, so, of course, we are all very interested in units of speech, right? So, such as phonemes. And when you look at those, this is just a similarity matrix between the phonemes. And you see that during the adaptation period, when the brain is still noisy, all the structures of phonemes are destroyed. But after that, they are nicely recovered. Some examples of that also are shown here. These are the average spectrogram of several phonemes. Yeah, I'm sure you recognize all their nice features, right? The zero of the nasals, the vowels, formants, the plosives, the stop and the release of the energy. And during adaptation, all of those features are gone, but after the adaptation is over, all of them are nicely restored. Right? So this is interesting, and it makes a prediction, right? Basically, the prediction here is that if the background changes for a few hundred milliseconds, you're not going to hear as well, right? So your perception should also follow what we see in the neural data. And we wanted to test that, but obviously it's very difficult to test that with continuous speech. So we put together a task that the background noise is exactly as we had before, but now we have consonant vowels, right? Like ba, pa, ta. Some of them, shown in orange, are placed immediately after the transition to a noise, and some of them in blue are placed after the transition is over, right? And then the prediction will be that your perception of the orange ones will be worse than the perception of the blue ones, right? If this adaptation actually is happening and that's what we see also in the behavior. You can break it into different conditions, and not as much in jet, which is less overlapping with speech, but in city and bar, it's very, very clear. And it's also surprisingly present in clean, right? So these two sounds, there are two consonant vowels in clean, right? There is no noise, but your perception of the first one is more degraded than the second one. And if you remember, in the neural data, we also had that rebound, right? Like when you go to clean, there was a rebound. So maybe some time that it takes neurons to come back from their adapted state. All right. Now, there's this question of, is this just a bottom-up thing, or is it the top-down, right? Like, is this because these people are listening to speech, or is this something that just happens because of the properties of the auditory system? So to test that, what we did in this case was we designed a very difficult visual task, right? And we asked people to solve the visual problem while we are playing them speech. And you can see that when they are distracted, their comprehension of the speech really is degraded. But the question is, the adaptation that we saw, is it going to be different in these two cases? And very interestingly, we didn't see any difference between the adaptation. So this mechanism doesn't seem to really require attention, right? It seems like just to be a signal processing property of the auditory cortex, right? That takes more modulated sounds and separates them from sounds that are not as modulated. All right, so, if you remember, I showed you that these different noises adapt in different locations, and we can quantify that, and that seems to be true. Like, jet noise is really, the biggest adaptation is in deep areas, speech is more on the surface. And this is not very mysterious because, these parts of the brain, they have different tuning properties. And you can actually explain the pattern that you see there by just looking at what frequencies, what temporal modulation rates these areas are more sensitive to. Okay, so this is interesting, right? And what I'm showing you here is one of those cases. You see the adaptation, and in this case we had the electrode in this area that that transient response adaptation was mostly present here. This structure here is called the planum temporale. This is the Heschel's gyrus. This is the planum temporale. It's also a secondary area, meaning that it's also deeper in the neural network. Now, here is one thing. If you put an electrode in someone's brain, right, you are not limited to just recording the sound. You can also zap that part of the brain, right? So that would be like, if you're looking at a neural network and you're just looking at the activity in one of the nodes, right, the activations of the nodes, but you could also just manually change some of the activations and see what happens to the rest of the network, right? And you can also do that in the brain. And it's actually done routinely in almost all cases. So every time a neurosurgeon, discovers that, for example, this area is the problem area, right? That's where the seizures are coming from. So their plan, ideally, is to go and completely take out that part of the brain, right? Really scoop it out. And that, removes the epilepsy seizures in many cases. But of course, there's also a risk, right? What if that area is very important, for example, for speech? So what they do is that they stimulate all of these electrodes, and they try to see whether an important function is impaired, right? So this was a case that we were doing that, and we saw something really interesting, which is every time one of these electrodes was stimulated, the patient said, I hear a sound, I hear a tone. Every time we were stimulating one of these electrodes, the patient says, all the background noise disappears. I only hear the voices. Right? And I have a small video of that here. Hopefully it would work so that you see this magic for yourself.
Okay, now I'm going to count to 10. 1, and just tell me when you feel a change and what it sounds like. 1, 2, 3, 4, 5, 6,
4.
7, 8.
Oh, 5. Oh, it's getting stronger.
What would you feel?
I can hear your voice more and more clear.
You use some other description. She's NPO right now, so I don't want you to talk too much because I don't want your mouth to get too dry, but you described it. What's the best way you can describe this in 10 words or less?
When you're listening to music and you're trying to adjust the equalizer, the tone, how you hear the music, easiest way to explain it.
Oh, wow. Would you describe it as, that person comes closer, everybody stays there where they are, or no?
No, it's as if someone just gives that person a mic and just... It kind of like they tell everyone to hush, to turn their background noise down, and let this person talk. The family likes fish.
And how was the quality?
5.
Five.
For me hearing it?
Yeah.
5.
Wow. Can you say more? Like why, what?
It's as if you're talking to me right now. Like everything in the back. I heard the noise that started with it, and then as soon as whatever you did, all I heard was his voice.
Just the voice?
Yes.
Family likes fish.
Mm-hmm.
All right, so nice method to do speech enhancement, huh? All these neural networks you're working on. Just a sticky electrode. All right, so is this real? Well, we put together a task, right? A standard speech intelligibility task. And we played the person in 2 conditions when that electrode were stimulated and then in the sham trial that there was no stimulation. And surprisingly, you see that the perceived quality and the intelligibility of the speech is significantly improved as that electrode is being stimulated. Obviously we have no idea why or what is happening or what is the circuit, that allows this sort of effect. But it's definitely very thought-provoking. All right, this was the first part of the problem, that we have a noise that is relatively stationary, and we see that signal-to-noise ratio is improved, speech perception is improved, and it seems like it's a bottom-up, so attention is not necessary. But obviously, let's talk about the most difficult case, that this could not work, right? If the noise is another speaker, then no kind of bottom-up process can separate the speech from noise. The 2 speakers, right? You need to have some kind of a dynamic. And this problem, I'm sure, is familiar to many of you. You have here the spectrogram of a mixed speech. And if you don't know what belongs to who, this is, of course, a very difficult problem, right? And this is the problem that our brain and also our machines have to solve, that they need to figure out what belongs to who. How do we color this? All right, so we do the same thing. We have, this, by the way, so this is based on a work that we did a while ago, that, if you have 2 speakers, you play them to a person, and you reconstruct from the brain of the person. Obviously, you get something that looks like that, you get something that looks like that. But if you play them at the same time and ask the person to pay attention to one or to the other, then interestingly, the things that is reconstructed from the brain only has the voice of that speaker. Right? If I put them on top of each other, these 2 signals were from exact same mixed audio. The only difference was the attention of the person. So we knew that this is happening in the brain, right? There is a level that you only see the representation of the person that you're attending to, right? So speech separation is real. It's in the brain. But we wanted to go beyond that, because this doesn't really say much about how it's happening. So this also has been shown with EEG. You see that early components are very mixed, and later components are more separated. And speech takes time to go through the different synapses, right? So the later responses, you can think of it as also deeper layers of the brain network, right? Because going from each layer to another layer would take some time, right? So later response means deeper in the network. All right, so we wanted to know how these shallow areas, primary and non-primary areas, are responding to mixed speech. And what is the interaction, right? What is the computation that happens between these areas that allows a person to separate one speaker from others? And there's also this notion of auditory objects, right? So these are like visual objects that, like if you have an object like this, like if I occlude a part of it, you still know that this is an object, right? You can fill in the blanks for yourself. So this notion about is there a notion like that also for the speakers? Like, is there an area that responds to a speaker as an object? So these are the questions that we wanted to address here. The experiment was quite simple. We have single talkers, and we have the mixture speech. In this case, to make it easier for the patients, we have a male and a female speaker. There's no spatial separation. This is all just single loudspeaker. And the people can do that very well, with accuracy more than 90%. All right, so the areas we're going to look at are, again, the same. We're just going to inflate the brain now to make it easier to look at them. So you have the primary areas. So every time I say HG, think of a primary, low-level area, right? Every time I say STG or orange, we're talking about these higher-level areas, right? So this is... All right, so what do we see? Here's an example from that higher area that, whenever... So in solid lines, you see the response to that speaker. The blue is response to that speaker. And these curves on top of it are the responses as the person attend to one or to the other. Right? So I hope you can see that the response really changes to match to match the response as if that speaker was alone. This other electrode, it's an example electrode that it doesn't seem to really care about who you're attending to. It just represents whatever it wants to represent. Right? So we have these two types of responses. One is modulated by attention. The other one doesn't care about attention. It just seems that it likes one speaker and it just responds to that speaker and doesn't care, right, about what you're trying to do. So we wanted to quantify these and see if there is a pattern in the brain. And one thing that you can do is to just take the single talker responses and look at their joint probability distribution, right? And if it's shifted to one side, for example, for that first guy, that electrode seems to respond more to speaker one, right? The second electrode, it's shifted upward, so it seems to respond more to a speaker two. You can quantify that, and then what we see as this property, like this preference for a speaker, is much stronger if you are in the primary area, right? And as you get to the higher areas, that speaker preference seems to go away, all right? So again, think of it as a neural network in your brain, right? And the shallower parts, they really care about the speakers, and they respond to one speaker or the other. As you go deeper and deeper, that property seems to disappear. By the time you get to STG, they respond not very preferentially to a particular speaker in this case. And it's also just to say that this preference seems to really come from their tuning property. All right, so like this is the average tuning property of those sites that respond to one speaker or to the other, and they really have the right features. They just happen to like a certain format frequency, a certain pitch, and as a result, it's just naturally these two speakers fall on this continuum, and one set of electrodes respond to one speaker, another set of electrodes respond to another speaker. All right, now if you look at the attentional modulation that I mentioned to you, from these four conditions, we can put together an index that the higher this index, the more this electrode change its response with attention, right? Like depending on if you say pay attention to that speaker or that speaker, if the response really changes to match the response to that speaker in isolation, this index will be high. Okay, we call it attentional modulation index. Now, if you look at the distribution of this one, it's the opposite of what we had before. Now, if you are in the early areas, the green areas, lower levels, they don't really care about attention. But by the time you get to those higher areas, now they really care about attention. And that also can be shown as their joint distribution, right? So you have these curves that all the green ones are in the primary, or the orange ones are in the secondary, and these guys care a lot more about attention. Those guys care a lot more about the speaker, right? You can also see it here that they go from blue to purple on the brain. So there is this really nice difference between these two brain regions, right? Like if you take these two parts of your brain, one of them really is just fixed. I said, I like these speakers. I don't care about attention. Don't worry about top-down. I represent this speaker or that speaker. And then there is another part of your brain that really is very dynamic. It says that, I just change whatever depending on the task, right? If you suddenly pay attention to something else, the response of that region is going to change. It's, it's interesting. I mean, it's very hard to make connections, of course, right? But this reminds me of feature extraction, feature summation parts of a network that, you create a nice representation and then you can read from that whatever you want. And that's actually what we try to test here. From that early part of the brain, you can test, and it's possible to decode each of the speakers very easily. And it doesn't really matter who you're attending to, right? So this decoder learns to decode the blue speaker, and if the person switches attention, it doesn't matter, even though it hasn't been trained on the other speaker. And if you try to predict those secondary areas from the early areas, these are the regression weights, linear regression weights that are given to those electrodes, right? So think of these as producing the feature extraction part of the representation, and these are the feature summation, right? And you see that these two areas are really seem to be, working together in that way, and you see it here for all electrodes. All right, how these weights are changed, right? How these secondary areas read out the speaker really depends on the speaker selectivity of those regions. So if you have an area that was responding mostly to one speaker, it also changes very, in a certain way to enable the higher area to read that speaker. Skip that in interest of time. Let me tell you a little bit about this auditory object concept, right? So this is, as we discussed, in the visual domain, we have this concept of objects that, if partly something is occluded, our brain still perceives it as one thing. So is this true also for speech, right? So if you have an overlapping speaker that sometimes completely masks the other speech and sometimes not, if your brain is insensitive to that, that's an indication that you have a concept of the auditory object in the head, right? Like there is this representation of a speaker A, and it doesn't matter who else is talking, how loud they are, right? I have this solid representation of that speaker. But if it's not the case, if it's also changes depending on what else is in the environment, then it's not really a very strong and compelling evidence for an object, right? Because it's still being related to other things. So fortunately, in a speech, it's actually quite easy to test that, right? Because if you have a speech and let's say this particular site responds to that feature, naturally, sometimes that feature in mixed audio is very overlapping with another speaker, and sometimes it's not, right? So if we get this variable degree of overlap for free, so we can test that auditory object concept. So here is responses to single talker, right, from one of the speakers, and and these are the times that sometimes the response to one speaker is greater than the other, so you could tell that this speaker is masked by that speaker. Here, this speaker is masking that speaker, and here they are masking each other similarly. So if we look at that as a joint distribution, you can then superimpose on top of that the responses to multiple talkers, right? And this will be your representation. So you see that when you're attending to this speaker, you have this gradient of colors, and it switches attention as you're switching switches direction here, right? As you attend to the other speaker. All right. So if I put this on top of each other, right? Rotate one and put it on top of each other, the rows and the columns of this matrix are very informative, right? And why is that the case? It's because if you look at the rows, it basically tells you that the speaker that I'm interested in, right, if it was alone and if it was mixed with another speaker, what is their relationship? And the colors here are the amplitude, the loudness of the other speaker, right? And it doesn't seem to care in this area, right? So that other speaker, no matter if they're quiet or they're loud, they don't seem to show up in the response at all. If you look at the rows, the columns of that matrix, you see the opposite, right? Now the response of that electrode to the unattended talker is always flat, right? And it just changes with the magnitude of the attended speaker. So this really shows that in this electrode, at least, there is evidence for an object, right? Because the masking doesn't affect how this electrode responds to the target speech. This electrode, on the other hand, doesn't show that. It has a very linear response. And the interesting thing is, if you look at the primaries and non-primary areas, it's again another indication that this idea of the auditory object of representing one speaker as a whole only appears in the secondary areas. In the primary areas, they don't really care. They respond to whoever is louder, right? So this is also very interesting. This is actually the first evidence that we do have this formation of auditory objects in our brain as we are listening to mixed speech. And another way of thinking about it is that in the primary areas, it's a very linear response to the speakers. But when you go to the secondary areas, one of them is non-linearly warped. And it's not just the gain, turning down the gain, right? That would be, again, a linear suppression. It's a very non-linear one. All right, so what I showed you is that we have the primary area that doesn't care about attention. It has a diverse tuning. It really cares about representing speakers. And we have the secondary areas that are very modulated by attention and nonlinear suppressed the unattended speaker. So for the last few minutes of the talk, I just wanted to talk about some more fun projects that we have. One of them is this idea of the decoding, right, that I mentioned to you, that we have the responses and we want to turn it back to sound. And obviously linear is where you start, but you can make it a lot better. Why is this important? When you can think about it as, see, a lot of times when we hear something and when we imagine hearing something, the pattern of the neural activity is very overlapping, right? So the question is it possible to say something about what the person heard or wanted to hear, right? And if it works, it could have possibilities such as brain-computer interfaces, right? So you might be enabled to restore speech to people who are not able to speak. And if you know who the person is trying to pay attention to, then you can think of a scenario that you have a hearing aid that is, people who have hearing aids don't use them because as soon as they go to a crowded place, it amplifies everyone, right? And then they are pretty much useless. But there's nothing that a signal processing algorithm can do unless if they know which speaker is target, which speaker is not, right? So is it possible to read that piece of information from the brain directly? And then that could direct a speech separation method. So I'm just going to show you very quickly a few results on that. Here is our speech reconstruction. It's an inverse mapping. There is possibility for speech neuroprosthesis. And the barrier so far has been that the quality of the speech has been really low. So, for that to work, we need to have a way of making it more intelligible. So, what do? Of course, we replace the linear method with a deep neural network, right? And you train it on all the possible frequencies in the neural data. And what we try to do is to reconstruct the parameters of a vocoder. It's a word vocoder. And we compress that using the autoencoder network just to make it more robust to noise and also lower the dimensionality. So here's some examples. This is what we had before with the linear model. Not very intelligible, and this is what we get with the DNN and the vocoder.
4, 5, 6, 7, 8, 9.
Yeah, that's. I hope you could tell that there were numbers. So it's much more intelligent. That's really cool, right? So this is like you don't know what the person is hearing. Like they have maybe a cell phone. You just see their brain activity, and then you can reconstruct the sound that they heard. And the numbers or the speakers were not in the training set. So this is all generalizing to unseen data. And this idea of the hearing aid also, now that we can reconstruct from the brain and we can reconstruct the attended speaker, is it possible to use that to guide a signal processor? Obviously, nobody wants to have implants if they want to have a hearing aid, right? And that, there's a lot of research on that people are trying to do it with minimally invasive methods or with around the ear EEG, and also even with just in ear, right? So you will have a couple of electrodes on your earphone, that is, recording enough neural activity to just extract this one bit of information, like who is the target. All right, so obviously, one problem with that is that if you want to compare the brain waves to the sound sources in the environment to figure out who is most similar, in reality, you don't have the separated sound sources, right? You just have the mixed audio. So in other words, you have to first solve this problem in machines. And that's what we've been trying to do, and if you have that method, if you have a nice speech separation, then you separate the sounds, you compare them to the brain, and you just amplify the one that is most similar to the brainwave, right, as the target speech. So this speech separation is something that we in our group have been working on lately, and especially with the focus that we want to have a method that is causal. It can be implemented in real time. And we had the deep attractor network, and recently we've had this time domain audio separation network that not only it is causal, it can be implemented in real time, but it also works really well, and it passes the ideal binary magnitude masks. So let me go to the demo. So now we have a way of measuring the brain waves and separating the speech. Let's look at the demo of this system. So that's what you're going to see. There is a box that takes the brain input and one channel of audio, and it's going to amplify the speaker that is most similar to the brain waves. Right? In this time, the person is focusing on the male speaker and then switches to the female speaker, and we turn on the system at 10.
How to be a shepherd. Dogs are great companions for a variety of people.
Especially when you know how to do it.
Have you ever thought about having your own
Shepherds
companion?
can be found all over the world.
If you do not have a dog yet, you need to understand the difference between breeds before making a choice. The breed of a dog can have significant impact on its personality, as well as its size, health, and required maintenance. For example, long-haired breeds, like collies,
move to Idaho or Wyoming.
require frequent brushing.
Buy a sheepdog.
If you do not have time
Some
to devote
sheepdogs
to
are better than
weekly or
others.
even daily maintenance,
You would do best
may
to
need
look on a
choose
dog
different
market
breed.
online or see
The
if you can
American
find a breeder.
Eskimo is bright.
Yeah, so that's pretty nice, especially if you have somebody who has hearing loss, right? For them, listening to multiple speech is much more difficult. But even, for someone with normal hearing, imagine, if you go to a crowded place and you can hear your friend, without other people, it's much nicer, much less tiring. All right, so in summary, I just showed you some of the dynamic and adaptive mechanisms that we have in the biological neural networks that enables us to filter out the noise and other speakers in the environment, and the result is a communication signal that is very robust and enables us to build cities like New York. Right? If our communication wasn't so good, we wouldn't live in a place like that. But the implications that it has for assistive hearing technologies, such as speech neuroprosthesis or for hearing aids. And I want to acknowledge all the students and postdocs who really did this work, James, Bahar, Yi, and Kang in particular, and our fundings, and thank you again for the invitation and listening.
So do you have any question?
I have maybe, it will be a very basic question, but you were talking about the electric potentials, but from my basic knowledge, there is also like the chemical stuff going on in the brain. Does this interfere with the listening experience, or that plays?
Chemical what? Chemical?
The chemical processes like neurotransmitters and stuff like that. Okay. Does this play a role in listening, or is there some research on that?
Well, I mean, that is like how this sort of computation is implemented, right? But when the neurons communicate with each other, they actually do send electrical signals, right? That travels through the axon, the output of a neuron. But when it reaches the input of the next neuron, the synapse, the transmission there is not electrical. Like there is no electrical connection between the two. So the electrical signal comes there, releases neurotransmitters, and that opens the gates on the other one. and changes the potential inside that neuron. So, it's an electrochemical kind of connection between neurons, and if you talk about like that level of implementation, like there are models of the simple junction that have, tens of parameters. Because every one of those gates, they have dynamics, they have properties, they have refractory times. So, so people who are interested in modeling that process definitely take all of that into account.
More questions?
Thank you for your talk. You showed a picture where you showed that different regions in the brain react to different types of noises, right?
Correct.
So I'm wondering, is that mainly hard-coded in the brain, or is it learned? I mean, does it depend on the person, on the experiences, or is it just like a hard-coded part of the brain that attends to different noises?
Yeah. So the thing is that the functional properties of the human brain are not very well understood. Meaning but on the other hand, there are some properties that are very much shared. Like if you look at the Heschel gyrus, that first part of the green part that I showed, right? And if you just play tones with different frequencies and measure the activity in different parts of that, you see that, there is a map that generalizes across subjects. Like those very deeper areas, the areas that are deeper in the brain are more high frequency. The areas that are closer to the surface are more low frequency. And that is just the hardwired map that, is pretty consistent across subjects. Although there is a lot of variability, like there are threefold variability from subject to subject in terms of those maps, but those sort of big picture sort of, like maps, they are pretty consistent. And the thing that we saw there that, like jet noise was more in the deep area, it's just a high frequency sound, and those deep areas are more tuned to high frequencies. And so this match resulted in that localized activity to jet noise. Yeah, so, but the short answer is that there are some hardwired things, but also all the synapses are very plastic and they change with learning and task.
Over there.
Thank you for the speech. I have a question. So, does the result of the test will change on the spectrometer? Will change if the person is able to understand what the sound is, like the semantic, or if the person is not able to understand, like if it's not a language, or something.
I don't fully understand the question.
Oh, okay. I'm just wondering if the result of the test will change if the person is able to understand what the song is, like the semantics. So if he knows the language or the speech or something, and or if the person doesn't know what it's hearing, is hearing the sound but he doesn't know what it means.
Yeah, that's a good question because like everything I talked about was very acoustic, right? But of course. And I talked about the phonemes, but you could go beyond that, right? That you have now syllables, right? You put phonemes together, and then you put them into word that also carries semantic information. And there is evidence that all of that information is encoded, and we also have ongoing research on that problem. That like how are those information extracted? In what I showed here, we haven't really looked at the contribution of the semantic. But people who do behavioral tests, they have shown that like in a cocktail party that is in a foreign language, you have a more difficult time. So definitely that kind of top-down knowledge is helping you to focus on one speaker and to filter out other speakers. And presumably, to fill in the information. Maybe some parts of the speech are masked, you don't get them, but with the top-down knowledge, you can fill in the blanks.
So the part where you shown that what the brain does is like this inattentive separation of all the sources in the signal and then cherry picking the one we want to see. It's very eye-opening for me because I always thought that it's like a PLL loop. From the lowest level you tune to whatever you want to hear. But if you are trying to separate into different sources, there has to be a limit on how many people we can separate at a certain time. Or so do you know this limit, or is it like, I don't know, 5 people, 2, or maybe 2 people and noise?
Well, I mean, behaviorally, that's easy to test, right? And a lot of people have done behavioral tests that, like then if you eliminate all the cues, like if you put so it depends on how many degrees of separation you have in your sounds, because if you have, the test that people have done, like Douglas Brungart is like one of the people who tested pretty much every possible scenario, that you could have you can add same speakers, you can add a speaker to itself and play to the person, and like that would be already very difficult. You have the same gender and that would help you a little bit. Different gender helps you even more. Do a spatial separation helps you even more, right? And so how many can we do? It really depends on the situation. I know studies that have shown even up to 4 speakers, people can still do separation as long as it's short enough so they can play it back in their short-term memory. But I think that the general thing of the question, I think, yeah, it is very interesting that just one clarification. I didn't say that the early areas separate the sound. It just, there seems to be this representation. Maybe we can call it a good embedding of the signal, right? That has this nice general property that different sources are easily separable, right? It's just because it has tuning, so many different degrees of tuning dimensions, right? That sources just naturally fall into different parts of this embedding space. But what are the limits of that? That's a good question, and we haven't really explored that.
So thanks a lot for the talk. It was quite interesting. So I have 2 questions. The first one is related with the variability of each person, basically. Could you study that differences, or do you have enough, samples to study that?
Variability of each person?
The variability of those areas that you are studying, if depends or not a lot in the different subjects that you are using for the experiments. Basically, in the same way like we are trying to, detect different phonemes or something like that for different speakers, and every single speaker has this kind of pronunciation, accent, or whatever. Yeah. And this is what we call like the variability of the speaker. Yeah, right. If you find also that kind of variability in the brain, on the
yeah, that's a very good question. Also relates to those higher level units that we abstract from speech, right? So you talk about like phoneme, for example, or phonetic feature representation. That implies that like that allophonic variation has been normalized, right? Now you've normalize all the possible ways you can say a certain phoneme into one category. And there is evidence of that people have shown that there is this categorical representation of allophonic variations in the brain, but the evidence, I think, is still mixed. You find some people who argue for that, and with different methods, like with regression methods, with just directly looking at that, with creating synthetic stimulus that goes from one continuum to the other. And then you can tell that, you can see that the neural responses change at one point categorically. So there is evidence for that, but I would say that that problem is not done yet. Like we haven't really studied it enough.
And sorry, can I go for the second one? And the second question is related with the experiment that you were showing there, where we were with the syllables. And we were able to be better in the second syllable than in the first one. Yeah. And you were showing specifically the case with clean speech. Yeah. There was also a difference, right, between the first one and the second one. So what is your hypothesis on that thing? Because, of course, you don't have right there any, adaptation to, because you are already in a clean environment. And the second thing is, like, if you subtract that difference in the clean scenario to the noisy scenarios, are we going to see still like a difference?
If we do what?
If you subtract basically the results that you are having, the difference that you are having in the cleaner scenario, I don't think it's this one. Is the other one with the bars? With the syllables and the bars?
Yeah, but I wanted to show you this, that, here is a case that, this is from the brain, right? And here is a case that the noise disappears, and you have this rebound, like the neural responses seems to, like the noise is gone, but you have this transient response. And you could also see directly in the responses themselves that, like every time the noise disappears, you have this pattern, right?
Okay.
So this could be, it seems like this transient response has a behavioral consequence also. It degrades your ability to perceive.
Yeah, so, but in the clean, as we can see in this curve, is like, there is no like this kind of peak that you have in the noisy one. And you have a difference also in the clean environment, you know, distinguish the two syllables. So that difference between those things, that interval between the first syllable and the second syllable in the clean environment, if you subtract that difference for the noisy environment, are we going to see still like a difference?
I see what you mean. Yeah, yeah. So we have done that with so in the neural data. And it's maybe a bit confusing because I have two cleans. Right? When I say clean, this is the clean in the noisy stimulus, right? This is when noise stops, it's clean for 3 seconds and noise starts again. But we also have a situation that we just have no noise at all, right? This is the exact same speech material presented to the subject with no background noise. And this is where I show him here that, you know, there is a transient and then these two become similar to each other. So for all the effects that we show here, we do compare that to when you just had no noise. Because your concern, I think, is that there could be something in the speech at that time that just caused that to happen.
Yeah, or something like, or our brain is like acting kind of capacitors somehow, and in the first C-level, you're really like not paying attention
to that. I jumped up on the
suddenly...
Yeah, but you know, in the behavior, I'm not worried about that here, because you know, here we don't have any speech anymore, right? So you just have this noise. Right? And you know, we have, this is like very long, of course. So people are listening to 15 minutes of this, and we have hundreds of those kind of syllables, right?
Okay.
And every, you know, like, so you have a lot of situations that there is clean, and you have the first one and the second one, first one, second one, and this happens many, many, many times. You know
so that is just a continuous waveform, I guess, and the and the subject is continuously like, you know, coming from jet to clean to city to what?
Yeah. Okay. So it's a difficult task. So they have to very quickly just say, I hear ba, I hear ta, I hear ta.
Yeah.
Yeah.
Okay.
Thank you for the talk. During the presentation, you have shown that it is possible to reconstruct the speech signal from the brain activity, basically, right? But the reconstruction is good, but it's not perfect. So do you think that this is because we don't have a lot of data for this problem, or you think that is because at that level of the brain we have high-level information and we are not able to reconstruct perfectly the low-level information?
Yeah, that's a good question, and I think it's probably a combination of both. And you know, one more thing to keep in mind is that our recordings also are these relatively large electrodes that you put in the brain, right? And each of those are summing the activity of thousands of neurons around them. You know, so if you, you know, like the auditory cortex has like millions of neurons, and we are, you know, recording like let's say 60 points, right? 60 averages of all those. So it's very hard for us to separate these two factors, you know, because our measurement is not at a resolution that can separate these two hypotheses. But one thing that we have seen, of course, if you have a better model, if you have more data, that always helps improving the intelligibility of the reconstruction.
So just another quick question. So you have seen, we have seen that okay, so if you add this external electrical signal to some neurons, you may have some better, the people feel that they can recognize the voice much better, right?
Yeah.
So my question is why this doesn't happen naturally? I mean, why the natural evolution of the human have already discovered this? So have any intuition about that?
Like if we have this thing in our brain that does that, why are we not using it? Is that the question? Yeah, so it's an interesting question. I don't really have a good answer for that because, you know, I don't really know how to make sense of this data yet. You know, one thing that I didn't show is that this effect that this person reports is temporary. You know, like, so you have this stimulation, the noises disappear, and they gradually come back. You know, so like one way that I just try to imagine what is happening, and I don't know if that's the case, is like, there is a over subtraction of the noise at that point? You know, and of course it comes at a cost also, right? Like if you're able to completely suppress all the background noises, you may not hear the tiger that's coming toward you. So you know, even evolutionarily, like it may not be in your best interest to completely get rid of everything other than what you want to listen to.
Okay, thanks for the talk. It was really interesting. I was wondering, all this mechanism you talked about in the brain, are they something we learn, or does a baby is born with it?
Which one?
All of them, the speech separation and recognition. Is a baby born with it, or is it something we learn as we grow?
Yeah, so that is not something that I study, so I'm not an expert in that area. But the first part of the talk that I showed you, that mechanism that allows you to separate, like, let's say, jet noise or street noise, that we also have seen it in ferrets, and people have reported also that in birds. And in the original paper, we had a computational method of that was very much based on just the properties of neurons. Like you have synaptic depression. Like when you have too many neurotransmitters, your neuron just shuts down. The neurons regulate each other. So those very simple just properties of the neurons explains some of what I showed. But of course, when you talk about the higher level things like your recognition or separation of speech, recognition is definitely something that you have to learn, right? And people have shown that there is this, with the developmental, depending on what you're exposed to, you lose some of your distinctions, you emphasize some of the distinctions that you have in that language, right? So that's something that's definitely plastic, and it changes over the course of the development. Other people have shown. Speech separation, I'm not sure. I don't know if babies can do speech separation. It's a good question. Something to test, I guess.
A follow-up question to the question about the speech reconstruction based on what you measure in the brain. So since you seem to have trained the DNN vocoder, there seems to be a reasonable sized data set, I assume. So my question would be, has anybody tried to build a speech recognition system based on those features? Just to see if the information contained in the features is enough.
Yeah, we've tried that too. Yeah, we also have a paper that we try to directly decode phonemes and, even maybe put on top of that like some very rudimentary language model. And you could do something to some extent, but the purpose of that work, we chose direct reconstruction of speech for several reasons. One is that, if the goal of this is to use as a speech BCI, that allows the person to also maybe convey other type of information, like the intonation, emotion. And also it allows a feedback, right? Because a lot of the time you build a BCI for a person, like to control an arm, for example, with their brain, right? And it doesn't work very well, but the person learns to adapt to it, right? So we have this machine that learns how to control that, and then there's this closed loop. Then you can update your algorithm, adapt to the new brain state, and there's this co-adaptation. So, in this work also, like if you can imagine that you have a speech recognition, these discrete units, it's very hard for you, if you miss a phoneme, you don't know how to change your brain state to make it better, right? But if you have a continuous variable, like actual sound that provides you continuous feedback, it could have some benefits. So that's why we have been following both sides of this, because there's advantages in doing that. Of course, it's an easier problem because you can just, if that's your goal, you don't have to, reconstruct the speech. So we have been following both of these paths to see, which one would yield the better result.
All right, next question. All right. Yeah, thank you for the talk. My question is related to the noise experiments, where you had people processing speech in noisy environments with the jet in the city and then the bar. And you show that there's a sort of noise invariance in the neural response. But I'm assuming as you increase the noise level relative to the speech, there's a point at which the speech is unintelligible. I'm wondering if the neural response is somehow proportional to that level, to the relative level. And at some point, is there a threshold where there's just no response at all to the speech?
Yeah, that's an interesting question because we kept our SNR pretty high because we wanted to make sure that it's always intelligible because we didn't want to bring in those semantic and high level. We wanted to make sure that it's always intelligible, and then we study that. But other people have looked into that, and I know of at least one study. The first author is Ding, that they showed that if you change the SNR, it stays relatively constant up to extent, and then it collapses. And that is also the time that the perception of the person completely disappears. So there seems to be some kind of a...
So I have one last question. I will finish. So, the last part of your talk, you talked about the focus of a listener, for a hearing aid.
Yeah.
Okay. And if we get information from the brain, that's excellent for the hearing aid. But this personal devices like Alexa and Google Home, they're interesting in this problem because which one, for example, if people are talking and someone walk up Alexa or Apple or whatever, they need to listen to that person rather than the other ones. Yeah. So in the brain is only, like they're doing speaker recognition, because I think the other companies that might be doing is they try to recognize the person who do use the word, to wake up the device to trying to track it later. Yeah. Is the human doing the same, or maybe the machine is not doing the right thing as a human? So what is your take in that?
Like separation and tracking.
Yes, because what's human doing in this case? Is it doing recognition tracking, or if it's visual aid, or how do brain help that information? So it's
Well, I mean, any information that you give to the brain, it uses it. Like if you have lip reading, of course it would. Yes. It would. But it's a good question because I don't know if we haven't really thought about the tracking problem. Which is a big one, because, we also work a lot on the speech separation, just technology part of it. And it's always a problem of how do you, so you separate them, great, but how do you track from frame to frame or from block to block, right? How do you keep memory of who was the speaker one and how it's done in the brain? I don't really have a good answer for that. I don't know. That's if there's a nice experiment that, like you can think of that people are using for the technology part, we could also try to use the same thing in the humans and answer that question. And I tell you next year, if you tell me.
All right.
Okay, so let's thank our speaker again.
Thank you.